Hi, everybody, and welcome to another episode of How to Sell Drugs, podcast put on by Lucy. The object of this podcast is to discuss drug policy, culture, and business with an emphasis on harm reduction. We believe that abstinence and prohibition-only policies result in poor outcomes for society. Today, we are joined by Dr. Weston Nichols. Don't let his boyish good looks fool you. He is a highly intelligent person. And we're going to talk to him about a number of his areas of expertise, starting with his Ph.D., which was in nicotine. And then we're going to talk a little bit about his work as a hedge fund operator.
No, analyst.
Focusing on biotech stocks. So, Weston, maybe you can give a little bit of a deeper background, and let's just start with talking about some of your research on nicotinic receptors.
Sure. Well, thanks, David, for having me. Pleasure to be here. Sammy as well.
Hey, Weston.
Excited about Lucy Gum. It's really cool you guys are doing that. And really excited to see it, continue its, strong launch so far. So, to give you some background on me, I grew up in Virginia. Found my way to Caltech for grad school, where I ended up doing my Ph.D. dissertation on nicotinic receptors. And I specifically studied, there were kind of two main parts of the thesis, which were, one, this small protein called LINX, which is a GPI-anchored protein. It's anchored into the membrane of a cell. And I don't know if you know a bungarotoxin? Have you ever heard of that? So,
refresh me.
Yeah, sure. So bungarotoxin is a snake. It's a protein that is expressed in snake venom.
Any particular kind of snake?
Snakes that don't live around here. I don't know specifically.
Not the bungaro snake.
And so it paralyzes the prey.
Okay.
Right. So
and it acts on acetylcholine, or
it acts on the nicotinic acetylcholine receptor.
Okay.
And it basically makes it not open, which means that you can't transmit. It binds to the acetylcholine receptor. I don't act in addition to the nicotinic receptors.
Sure.
So the prey can't move.
Sure.
And
makes it easier to eat.
Yeah, real easy to. Yeah, move. Exactly. And so, the structure of LINX. So before I got there, there was a postdoc actually in the lab, Julie Miwa, who I think, actually I'm not sure if it was her, but anyway. So the structure of LINX is quite similar to bungarotoxin, okay. And so it was hypothesized. She hypothesized that it's essentially expressed. It's turned on at a time point during development of an organism, in order to kind of allow the nicotinics to fire and then stop firing, okay, to kind of stop working, which essentially what that allows them to do is to branch out and form lots of new synapses very early on in development, and then stop.
Okay, so this protein is typically found
all of us. So we all have it
or?
now. No, it turns on in a mouse. It turns on, I think it's something like day E14 or something like that. I forget if it's before birth or after birth, but very early on.
Okay.
And it probably reduces the amount of new synapses formed, because when you get loss of activation of nicotinic receptors, you form more synapses.
So that's interesting. We should delve into that part later.
Yeah, I don't have much else to say, but people think that, right? And so that's why nicotinic receptors are very important in learning and memory and mood and pain and lots of things. And there's been lots of work done to try to figure out how you can make a drug that would be an actual pharmaceutical product for lots of different indications. And
so a nicotine analog.
Yeah, exactly. But the hope would be that you bind to certain subsets of the nicotinic receptors and not all, and somehow you select the ones so you can specifically target, pain, or and let's come back to epibadidine, because that was kind of the reason for pain in a minute. But focus, learning, and memory, because we know that another key reason to delve into nicotinics from a drug development perspective is the drugs that work for Alzheimer's. They don't work very well. They basically help your focus, help your cognitive abilities they basically give you 6 months back. So it's not very effective, but they do something, and everyone's on them.
Do they slow progression?
No.
So they
don't.
just basically, wherever you are, you're 6 months.
Yeah, it basically goes 6 months back, basically.
And then if you stop taking them, do you fast forward wherever you should have been plus 6 months?
That's a great question. I think the answer is probably yes. I don't know. I'd have to look it up.
Sure.
But I think it's probably yes.
Okay.
You probably don't stop taking them because...
Yeah,
Why
I
would you?
mean,
Why would they?
some people stop taking for tolerability problems. So there are people that come off, and I'm sure this experiment's been done. I'm sure we have good data on it, but I just don't know.
Be sure to talk into the microphone.
You got it.
Closely.
You got it. Sorry. And so the drugs for Alzheimer's are acetylcholine esterase inhibitors, and so they inhibit the enzyme that breaks down acetylcholine.
Is that kind of like an SSRI? How it prevents like serotonin from being broken down?
It's kind of similar. SSRIs inhibit reuptake of serotonin.
Okay.
So
it's like an MAOI, basically.
Okay.
Like the analog of an MAOI, but for acetylcholine esterase.
But for my sake and those of the people listening, can we simplify the explanation for that a little bit? So you're saying an acetylcholine esterase inhibitor is something that stops the enzyme that breaks down acetylcholine.
That's right.
So
outside
that
the synapse.
In the synapse, yeah,
in the
within
synapse.
in the cleft, yeah.
which leads to a higher level of acetylcholine, the neurotransmitter, in the synapse, which means that and acetylcholine is an excitatory neurotransmitter.
Yeah.
So it enables these synapses.
Yeah, well, I don't know.
To fire
I don't want to
better?
say excitatory. I mean, because it binds the nicotinic receptor, and then it makes the nicotinic receptor more likely to open.
That seems excitatory.
Yeah.
Excitatory describes like the
Yeah,
downstream effects of the receptor.
Yeah.
Okay.
Like glutamate is the most
Sure.
is like the
canonical
prototypical
example,
excitatory, and then GABA is you would think kind of inhibition,
Right.
But it depends on what neuron it's on, because you do have nicotinic receptors on the GABAergic interneurons
Sure.
within certain brain regions,
Sure.
which decrease overall excitation in the brain.
Sure.
Yeah.
Okay, so let's zoom back out for a second. So acetylcholine esterase inhibitors help people with Alzheimer's function better because...
Well, I mean, I don't know that we know exactly.
But what do these drugs do? Like in terms of, they increase the cholinergic tone of the brain.
I think that's the theory.
Can you describe what cholinergic tone means, Sammy?
It's just, it increases the amount of activated cholinergic receptors in the brain because you have more acetylcholine in the synapse.
Okay.
Right.
Well,
So it's like
it's
a very
extremely
gross,
complicated and very confusing, because if you dump a bunch of acetylcholine onto a population of certain types of nicotinic receptors, such as alpha-7, for example, and there's lots of different types, right? But alpha-7, it desensitizes very rapidly. So when you have acetylcholine, two acetylcholines might bind the channel, and then it opens, but then it clamps shut, and it's in a desensitized state where it's shut and you have two acetylcholines bound. And so it's extremely confusing, because some people say, so whenever you do an experiment, essentially, with alpha-7, you can either say, oh, it's increased, you can say, oh, the drug increased, like acetylcholine or nicotine, increased alpha-7 function, and therefore that's what it's doing. Or you could say it decreased function through desensitization, and you don't really know which one's correct.
Sure.
Right?
Sure.
So, you can very hand wavy, and it makes it very difficult to develop drugs, because you can have drugs with different properties of how much desensitization they have. And there's actually one very exciting drug that basically gets around the issue of desensitization, because it's not an agonist. So an agonist, such as acetylcholine or nicotine, would bind the alpha-7 receptor, and it opens it, but then it desensitizes quickly and shuts, right? Alpha-7 is one of the fastest desensitizing nicotinic receptor.
Cool.
And
so it's like, I've done my job, I fired, and now I'm taking a long rest.
Right, exactly.
Okay.
And so there were a bunch of programs targeting alpha-7 with agonists,
Why would you want to target alpha-7?
That's a great question. So people thought that alpha-7 is expressed in certain brain regions that, such as the hippocampus and the entorhinal cortex.
Okay.
Yeah. So thought to be if you could increase brain activity in these regions, you could help Alzheimer's patients.
Okay.
Because clearly,
so
acetylcholine does this somehow.
That does sound like downstream excitatory, right? It sounds like then that perhaps people with
that's
that
the
have
thinking.
Alzheimer's. That
is the thinking. But what if acetylcholine esterase inhibitors work via desensitization of alpha-7?
So it could be that. So it could be two competing mechanisms, and we're not sure what it is. It could just be that there's too much gunk in the brain in the form of tau protein or whatever in Alzheimer's. And so,
I don't know. Yeah, I don't know. I don't really want tau and amyloid beta. The field's a mess.
Okay, so avoiding that. But it could be that in the brains of patients with Alzheimer's, they have a hard time getting their neurons to fire appropriately, and that could either be because they don't have enough of a jump start, or it could be because there's too much noise. And
I don't know. I really don't know. I mean, this is all very difficult. I don't know if their neurons aren't firing. I mean, they have less synapses in general, Alzheimer's patients.
So it could be,
but also old people.
Could be a number of things, but we have noticed the symptoms improve when you give them
so we do know that. These drugs do work to some extent.
Sure.
And so the thinking is, let's interrogate that as much as possible to try to find more drugs and, just make progress.
Sure.
And so we know acetylcholine esterase inhibitors work, number one.
Okay.
Number two, we know alpha-7 is highly expressed in brain regions where it seems that Alzheimer's kind of starts.
Okay.
Where you start to lose synapses in the entorhinal cortex and the hippocampus kind of before other regions.
Okay.
And so maybe if we specifically target alpha-7, we can have a good effect, and maybe it's a better effect than just acetylcholine esterase inhibitors. And so there was a very promising phase 2 clinical trial done, and I remember reading it when I was in grad school. So I don't know when it, maybe 2007 or 2011 or 12, it probably came out, and it was a company called Forum Pharmaceuticals. And really smart group of scientists, really cool company.
Because they were taking all these acetylcholine esterase inhibitors.
Yeah, I mean, they were really interrogating these nicotinic hypotheses.
Sure.
And then there was another company, Targacept, which was a spin out of, I think,
R.J. Reynolds.
Of R.J. Reynolds, right, of a tobacco company. And then AbbVie also had a compound. So they had these 3 compounds. They were all alpha-7 agonists. And they were pretty much all of them partial agonists.
What is a partial agonist?
Oh, it just means that when they activate the channel, if you compare them to acetylcholine, they let less ions through per unit of time. So we call that efficacy of an agonist.
Sure.
So it's a lower efficacy agonist. People get very confused by all these terms, partial agonist, but if you just define them clearly, it's very simple.
Sure.
So basically, we had 3 different partial agonists, and they were all a little bit different, but they seemed kind of similar. And then there was this one paper where the Forum compound, it turned out it was actually probably potentiating the channel at a certain dose, and that dose seemed to be where it worked the best. And it was an inverted U, right? So dose response. So basically, at lower doses, the drug didn't do that much, and then at this one dose, it looked good, and then at higher doses, it didn't do well either. It didn't do much either.
The inverted U is something that you see in dose response for many drugs.
I mean, you don't see it that much. Anytime you see an inverted U, you got to be very worried that maybe this effect is not real.
Interesting.
Yeah. Allergan, by the way, depression drug, just failed last week. They had a really strong inverted U in their phase 2. In the phase 3, they just failed like 4 trials, all dead negative.
Wow.
So anytime you have an inverted U, you gotta be
well,
worried.
Inverted U is really just like describing the shape of a curve.
Yeah.
So like what you should be worried about depends on what the axes of that graph is, right? Because
Okay, sure. That's fair.
I guess what you're describing is like it's the dose response is not linear, or it's not dose proportional.
Right.
And that's usually a red flag when you're talking about efficacy or something.
Yeah,
right?
totally. I agree. Yeah.
Okay, so to just zoom out again, inverted U, what we're talking about is a graph of an axis, which basically says the
x-axis is dose,
right,
and the y-axis is efficacy.
Is desired effect,
yeah,
efficacy.
Exactly.
So when you chart this upside down U, you're saying at a low dose doesn't really work very well.
Right.
At this medium dose seems to work fairly well.
Yeah.
And then at a higher dose stops working again.
Yeah.
And you would contrast that with something that would be a different kind of dose response curve. Let's say it were a linear curve, which is a little bit, little effect, medium effect.
Yeah.
And then the reason why you wouldn't just dose all the way to the right of the X is usually because patients can't tolerate it.
Yes,
they get
that's
side
exactly
effects.
right. And that's exactly what you see with acetylcholinesterase inhibitors in Alzheimer's.
Okay.
You push the dose, and you get more efficacy. There is somewhat of a ceiling effect, but there's no inverted U at all, right? And you start to get tolerability issues. So no one really goes that high.
And so is that because, and that could be a number of reasons. It could be the actual receptors in the brain tissue. If they're oversaturated, something is getting messed up there. Or it could be that you have acetylcholine receptors and other neurotransmitter receptors all over your body in different tissue types.
Oh, well, why you get tolerability problems?
Yeah.
Yeah, totally. Yeah.
Yeah, I guess if we could take a second and just zoom out, because I kind of want to get to the
drugs.
Yeah, because it seems like what we've described is basically acetylcholine esterase inhibitors, which broadly increase the activation of all nicotinic receptors in the brain, seem to have an effect for treating Alzheimer's disease. And that's like very, very surprising, because that's essentially the only type of drug that works for Alzheimer's disease. When we go and try to sort of become more specific and try to activate certain receptors, certain subtypes of receptors, which is when we're talking about alpha-7 or alpha-4 beta-2 or whatever.
Yeah, types of nicotinic receptors.
It doesn't seem like those drugs have worked.
Right, they didn't work. So there was a great phase 2, and it looked good. It worked at the dose where, it worked in mice, and where the inverted U shows that it works, and, it looked really good in phase 2. And then in phase 3, basically all these drugs failed. And, some had tolerability issues, some just didn't have efficacy. And they just didn't work at all.
It's a bummer, man.
Yeah.
And then you were describing that those were all agonists.
Yeah.
And you're saying there's potentially a new class of drugs which are partial allosteric modulators.
Just allosteric modulators.
Okay.
Positive allosteric modulators.
Okay,
So
hold on. We gotta define that term.
Yes, we will. So let's ask the question: what happened? Why did all these drugs fail? We don't know. So what are the reasons? One, it could have been that these things are activating the channel, but they're just not activating the whatever channels you need to activate, because we know acetylcholine would work, right? If you just inhibited the degradation of acetylcholine via use of an acetylcholinesterase inhibitor.
Sure.
So maybe it's not alpha-7, or maybe for whatever reason this drug is causing desensitization of the receptor. And so, if you actually were able to positively modulate the receptor, then you could have efficacy. But these drugs had desensitization. So maybe desensitization is the reason why they don't work. Mm-hmm. Or maybe this is just a bad target. Really, those are the two issues, right? One, maybe it's not a good target, and two, maybe it was desensitization, and so you're not actually getting activation of alpha-7. Mm-hmm. Maybe. Mm-hmm. And so there's this really interesting company from Australia, Bionomics, and they're partnered with Merck. So Merck is going to run this trial, but they have an alpha-7 PAM, positive allosteric modulator. So allosteric just means it binds at a site on the alpha-7 receptor that is not the acetylcholine site. And so PAM in total means that it binds alpha-7 and causes it to be open for longer duration. And it doesn't cause desensitization. Well, there are some PAMs that might lead to desensitization, but this one doesn't.
And does that mean you still need the primary agonist or an agonist in addition to that allosteric modulator?
That's exactly right. So a PAM is not going to open the channel all by itself. So that's very interesting. So now what happens when you combine the PAM with an acetylcholinesterase inhibitor? We'll find out.
So like what would be an accurate analogy if I'm bench pressing and you're spotting me and we're trying to open we're trying to get the bench press, to the height of my arm to open the receptor.
Yeah.
I'm the agonist because I'm the main force that's supposed to open it, and if you just help me a lot, you're kind of changing the ease
sure. Yeah, but there's more to it because the PAM can change all these different properties. It can change how long the channel's open. It can change how long it takes the channel to desensitize.
Right.
It can change the concentration of acetylcholine that is required to open the channel.
And it does this by
make it lower.
changing the shape of the receptor itself, usually.
Yeah,
right?
yeah, slightly, yeah.
So there's a lot of, this is a hard thing to visualize, but it's in a lot of biochemistry textbooks. There's really good figures out there. If you just Google
lots of
PAM,
figures. Yeah.
You'll be able to see a figure.
Yeah.
Okay,
yeah.
So moving on then.
So I mean, I don't know. I'm excited about this PAM. It's interesting. It's going to be fascinating to see if it works.
Well,
hopefully Merck runs a big phase 3 and answers the question.
It sounds like there's a lot of questions to answer about these PAMs. Like, what type of PAM would actually work?
And what
each
receptor?
of those is like in a different experiment, right?
Yeah,
That's going to require thousands of patients to understand if it's going to work.
And what receptor is this for? This PAM?
Alpha 7.
It's
the Alpha 7 nicotinic receptor.
So, how many different nicotinic receptors are there? Why alpha 7 all the time? Why alpha 7?
Because of the brain regions where it's located.
Okay.
It's specifically for Alzheimer's.
Okay, that's right. Okay.
Yeah, so nicotinic receptors, just high level, they have 5 subunits, and you combine 5 subunits to form one channel, which is essentially a pore in the membrane, because they can open and close.
Mm-hmm.
And the 5 subunits, they have various combinations. So in terms of just proteins, you have alpha 1, 2, 3, 4, 5, 6, 7, 8, 9, and there's a 10, and there might have been like an 11 discovered recently. I don't know that much about the real high numbers, but so there's at least 10, I think. I think the higher numbers are like in the ear only or something. But basically in the brain there is some number of them. So of the subunits, it's generally either 5 alphas, as with alpha 7, it's actually 5 alpha 7s. And then there's the most common receptor in the brain is alpha 4 beta 2, which has either 3 alpha 4s and 2 beta 2s, or it can have 2 alpha 4s and 3 beta 2s. And that was the second half of my PhD.
Okay, perfect.
So for that, there's been some hypothesis that nicotine could reduce your risk of Parkinson's.
That's exactly what I wanted to ask next. Okay.
Great. So, essentially, smokers, there's epidemiological data that smokers are less likely to get Parkinson's.
And controlling for the fact that they die earlier. They did control
Yeah,
for that.
yeah, they control for that. And so the question is why and what's going on there. So the alternative hypothesis that was, so it's kind of similar to the debate. I know you know a lot about the debate, Sammy, of does smoking cause lung cancer or not? Because that was a fascinating time in history. Obviously, it does cause lung cancer, but
no longer a debate.
Not a debate anymore. But it was fascinating to see the science develop, and I know you can talk about that a lot more. And hopefully, I don't know if you've already done a podcast on that, but that's a fascinating topic for another podcast. If I would much appreciate listening to. And so, but for Parkinson's, so the alternative hypothesis is that there is some factor that makes people both less likely to get Parkinson's and more likely to smoke for some reason. And that's not crazy. It could be true, right? So that would explain the correlation between smoking and lack of getting Parkinson's, because there's a correlation, right? But so how do we explain that correlation? So it's either smoking reduces your risk straight up, or there's some factor X that causes both, more likely to smoke and less likely to get Parkinson's. And we don't know what factor X is.
Could it be the MAOIs?
No, because patients, you start taking those if you already have Parkinson's. You're not going to start taking those early, at least as far as I know.
Aren't there MAOBs? MAO.
I don't think those have a link with increasing or decreasing your risk of Parkinson's. I think the factor X could be.
Selegiline, isn't that an example of something that?
Yeah, that's
take
MAOB, right? I think that's MAOB.
Yeah.
So but I mean, I don't think it causes Parkinson's. Yeah, because we need factor X to be something, or either way. Because I mean, it definitely helps Parkinson's patients, but we need factor X to be something that both increases your risk of smoking and decreases your risk of Parkinson's.
Why does it need to increase your risk of smoking?
To explain the correlation between the two.
Well, what if it's just something that's present in tobacco?
Oh, is it?
That would be the other hypothesis.
That would be the MAOB.
Oh,
No,
wait, is it present in tobacco?
No, that would... Basically, you're trying to separate causation from correlation.
Yes.
There is a correlation. You're trying to determine, is it causation or of tobacco or nicotine, or is it some other factor independent of tobacco, preventing Parkinson's, that causes you to smoke, be more likely to smoke, and be less likely to develop Parkinson's.
Right. So it could be some genetic thing that actually could make sense to me.
So the genetic thing would just be like, I fucking love smoking cigarettes because I'm genetically, and for the
because
same
you had
reason
some
why
sort
I
of-
love smoking cigarettes is the same reason why I'm not likely to get Parkinson's.
Yeah.
Or I happen to smoke cigarettes and either nicotine or one of the 7,000 other chemicals.
It's neuroprotective.
Yeah.
So there's trials underway to test this. There's one that's coming out very soon. I don't know the results yet, but I think they used a nicotine patch to try to see what effect that has on Parkinson's. And I mean, it was a big trial. I don't know much about it.
This
but
isn't Paul
it's
Newhouse.
coming out soon.
Paul Newhouse is Alzheimer's primarily.
This is the Michael J. Fox
Foundation.
It's a big European.
trial.
I think it's a big European trial.
Right.
Yeah.
And we don't know.
We'll get the results very
are.
soon.
Okay.
I think the trial's done, but I think they're just still analyzing the results or something. I haven't kept up with it, but it's coming out soon, so it'll be in the news and stuff. Hopefully.
So if you see that to be likely to be the case, will you be slapping nicotine patches on?
Oh man, I don't know. I mean, I suppose we really should, consider as a public health thing.
Yeah, I mean,
using nicotine.
We give
to prevent-
statins as sort of a prevention type of medication. I don't think that that's necessarily widespread and accepted, but it is something that public health officials are debating whether that should be done.
Yeah, I mean, if that trial's positive, that's a pretty strong case that that smoking does make it less likely to get Parkinson's, and it is the nicotine that makes you less likely to get Parkinson's.
Right.
So hopefully it's positive. I mean that would be. I mean, everyone should be taking Lucy gum every day. Is really what I would take away from that.
That would be my takeaway as well. I mean, and also using statins as an example, I think the side effects of long-term use of nicotine are pale in comparison to the long-term side effects of taking a statin.
Are there? I haven't looked into that. Are there any long-term side effects of nicotine? Just nicotine without and not smoking?
Not as far as we know. I mean, there's some theoretical data that it could be it's not a carcinogen, but there's some rat data that it could be it could help with angiogenesis of tumors. But we talked to a really famous scientist who's been studying nicotine for a long time. I only don't say his name just because I'm not sure how vocal he is about this opinion. But when we spoke with him, he said if you looked at the snus data, which they've been struggling to prove any correlation between long-term use of snus even and cancers, that you would see a increased rise in all cancers.
And
among
what
snus
is
users.
snus?
Snus is like those little tobacco pouches.
But it's
They've
tobacco.
But they're pasteurized. They're treated in a certain way so that it's lower in the nitrosamines, the sort of prime carcinogenic agent that we worry about.
Interesting.
And then the other long-term theoretical effect of nicotine use is because it's a short-term vasoconstrictor, that maybe it's
you get a small bump in blood pressure, probably.
Yeah, potentially. But it seems
that's also controversial.
Okay.
It seems like it might lower blood pressure chronically, but it does increase your heart rate acutely. And so because of the amount of nicotine that people will do during the day, the amount of time you spend in sort of tachycardia people think that might be a risk factor for cardiovascular disease.
Yeah, that's interesting. I mean, I think it's been pretty well studied. Caffeine has been pretty well studied for its effect on cardiovascular disease. And I don't think it has any increase in the risk of cardiovascular disease.
Right.
But there certainly are autonomic effects in terms of increased heart rate and blood pressure in the very short term. But those autonomic effects go away with continued use.
Right.
So.
And Paul Newhouse, actually, the professor at Vanderbilt who studies nicotine patch and other nicotine uses for the treatment of Alzheimer's, gives high dose nicotine, used to be intravenously, to very old patients and has not had any issues.
Interesting.
There's also been a lot of trials for NRT in people that have pre-existing cardiovascular disease or people that have had events in the past.
That's good.
And it doesn't seem to increase the risk of death or future events relative to control.
Yeah, that's pretty good.
All right.
Okay.
And because I've been stuck with 2 biochemistry PhDs for this podcast, we've been going really, really deep into the science. But I want to spend a few minutes talking about Weston's career path, and for lots of our listeners who may be interested in what kind of careers in science there are, and sort of how you got to where you are, and what is your kind of day-to-day look like, and can you describe that a little bit for people?
Yeah, sure. So, I graduated from Caltech, and I had a pretty good scientific, ability to evaluate research, and I was lucky that I was in a nicotinic lab, because at the time there were all these drugs that we just talked about, and so I was able to kind of gain by looking at all that clinical data, and had access to talk to the people at these companies, and so I kind of began to learn how to evaluate clinical data as human clinical data, and just kept on the path. One thing I do pretty much is a big core of my process in finding. So I just look for investment ideas, right? Mostly in public markets, privates as well, but mostly public. And you just look for stuff that's misunderstood, where you think the value is a lot higher than what's priced in currently.
So, for my layman's view on your process and how things work, it seems like you kind of look at 3 kind of buckets of criteria. The first one being the publicly available data that anyone could see, which is what are the company's financials, what has it done in the past, what's in the pipeline. Then you look at the trials that it has done on different molecules in that pipeline, and you can use your math abilities to basically see if you think that it's statistically underpowered or there's some sort of anomaly in the data. And then finally, because of your biochemistry background, you're able to look and say, what does this molecule look like compared to other molecules that may have worked or not worked, or what is a plausible mechanism for why this works? And so that's why you, as a biochemistry PhD, has an advantage over someone without that scientific background who's analyzing biotechs?
Yeah, I guess. I typically divide. I have lots of ways of dividing things up just to help me frame problems. But one way I do divide it up is the preclinical data, the mechanism of the drug, and then the clinical data. And if they all make sense, and for whatever reason the market doesn't think that it makes sense, then you look at the drug and say, okay, well, what % chance does this drug have of succeeding and of getting approved? And then you ask the question, all right, well, if it gets approved, what are the sales going to look like? And then you ask the financial question of, well, if it has those sales and all that stuff is right, or at least my estimates are conservative and I think they're going to be at least that, then how much money is this company going to make?
And how much will the stock price rise?
Yeah, exactly.
And then you also will sometimes even interview the leadership teams at these different companies.
Yeah,
right?
you meet with management, sure.
And that's something that all hedge funds do.
Yeah, everybody
in all
does
industries.
that. Yeah, everybody does.
But is there some amount of poker that goes on when you ask someone a tough question if you see the fear in their eyes?
I don't know. I mean,
obviously they can't tell you inside information.
Yeah, they don't. I mean, there's lots of regulations. I mean, they get in big trouble if they do that, and
right.
So they don't. It doesn't really happen that much. But yeah. I mean, I don't know. Most of my good investment ideas don't come from getting some kind of read from management.
Sure.
I mean, maybe. I
It's
can't
just the
think of an
icing on the
example.
cake, potentially.
Yeah, maybe, but it's just so easy to mislead yourself with that stuff.
Yeah. Were they blinking because they had something in their eye, or?
Yeah. I mean, I don't know. I mean, even if you know someone very well,
Sure.
You've met them, 50 times over the course of 10 years.
Sure.
I mean, I just don't think that's the basis for me personally for an investment decision. There are people that are very good at it.
Sure.
Okay. That's not me.
So let's talk about the biotech industry at large then. What subsets or drug categories do you see a lot of exciting movement in the next 5-10 years?
Well, I try to actually be really therapeutic indication agnostic. I try to look at everything because for a couple of reasons. One is you don't want to cross off indications you think are not promising just because, there hasn't been a drug that's worked.
Mm-hmm.
I mean, you're looking for indications where there is a real market, where there's a real need, where, patients would actually take the drug if it were approved. You are...
we've got some
friends.
rap music playing from our neighbors here. Hopefully we can edit some of this out. And we're close to being finished. So if your concentration hasn't been too broken, you're saying that there's no particular industry that you would want to call out in particular. You're saying
well,
that,
look, I think we're making progress everywhere.
Well, that's encouraging.
Yeah, because...
You never know what you're going to get.
You really don't. And, success comes out of nowhere sometimes. And
Well, let's talk about Alzheimer's in particular, because that's something where, historically, the great majority of drugs have failed. I think the failure rate for drugs in the clinic is over 99%.
Yeah.
What would make you want to invest in a company developing an Alzheimer's drug? What would it take to convince you?
Just the same thing it would take me in any other indication. The mechanism makes sense. You're not going to prove it with the mechanism in Alzheimer's because so few things are proven. The preclinical data, I probably would place less weight on because, if you're helping Alzheimer's in a mouse, I don't really know what that means, right? There are certain things that are going to translate well from mouse to human, where the physiology is very similar, but in terms of just in general, most cognitive indications, the mouse doesn't mean a whole lot. What you can say from mouse data is that the drug is active, and that's about it.
Unless it jumps out of the cage and starts rattling off Shakespeare to you.
No, yeah, that hasn't happened.
Well, so can you delve into why the mouse data is not relevant for Alzheimer's?
It hasn't translated before, and I don't know what it means. Right?
Well,
I don't know.
If a mouse suddenly is old, exhibits what we think are the mouse symptoms of Alzheimer's.
But I don't know what that is.
So can't run a maze. Can't learn.
No,
how to run
I don't
a
know what
maze
that means. So what are the tests that they use?
That would be indicative of some sort of hippocampal improvement, right? If you can go
No,
from
I don't know.
memorizing a maze to being able to memorize a maze.
I don't know. I really don't know.
So maybe one of the reasons is that mice don't they don't get Alzheimer's, right? A lot of human diseases, you can replicate the pathology in a mice by, for example, creating the mutations that are, that seem to be causative in humans, or by, infecting it with the pathogen that would also be infectious in humans. But with Alzheimer's, mice don't live long enough to get Alzheimer's. They probably don't even have the same brain structures which would lead to development of Alzheimer's.
Right.
So how do you even create a model that somehow replicates the symptoms and pathology of Alzheimer's?
We need to set up some sort of mouse summer camp in Florida where all of the older lab rats and lab mice can go to.
Well, they do have retirement homes for monkeys in Florida.
Okay.
Maybe there's some interesting thing. I'm sure there are very good scientists doing interesting things with new animal models of Alzheimer's.
Those poor monkeys,
I don't know enough
though.
about it.
They just when they thought that they were done and retired, it's like
no, they have
we've
good.
got-
They're
Just
treated
when they
very
thought
well.
they
They're
were
treated
out.
very, very well.
They pulled him back in.
That's so sad.
They live in like little apartments and they have TVs and stuff. It's really
The monkeys have
yeah,
TVs?
Yeah, it's. I've read a little bit about it. I don't
Do
know
they
that
play
much,
golf?
but no, they don't. But they're treated
do
well.
Zumba.
They're treated well.
Pretty
So
good
the
at
third
Zumba.
aspect is the clinical data. If the clinical data looked great for an Alzheimer's drug, then I would consider investing.
Okay.
But it's very difficult because if the clinical data looks great for an Alzheimer's drug, well, probably most people know about that.
Yeah, that's
So,
fair.
I'm trying to find something good in Alzheimer's, but it's very difficult. It's
Okay.
very difficult because it's very picked over. Everyone looks at it,
Yeah,
right? You want to look at things that other people aren't looking at so hard.
sure. So finding undervalued opportunities
Right.
is the main activity of a intelligent investor.
I
So
think
before we go, I think we'll have to ask you just one sort of parting question or series of questions. I think you have a really interesting perspective because you analyze the efficacy of drugs all day, and most people have a blind faith in modern medicine.
Yeah.
So, is there anything on Dr. Weston Nichols' list of things that most people don't know or don't consider the risks seriously enough in certain medications that they take, or things that they can do? Maybe it's a dietary thing that can really help them. Is there anything that you kind of think about yourself that you would recommend to your family that might be not well known, or maybe something that's well known but you would want to emphasize anyway?
I don't know that I have any particularly weird habits. I mean, I'd say lifting weights,
Okay.
and just making sure you're strong, because I just read a really fascinating paper in, I think it was Lancet, where it came out in the past 2 months. Being frail increases your chance of Alzheimer's about the same % as having a brain full of amyloid, which, some people think that the field generally, well, these did think in various states now, that amyloid causes Alzheimer's in the brain. There's a lot of failures, so maybe we have another couple big ones coming up here. The Biogen aducanumab antibodies data is going to come out next year.
Okay.
But just not being frail. But just going back to your one other thing you said, why it's important to look at many different indications for people who might be interested in this. I think it's critical to look at many indications. So I'm relatively early in my career, right? I'm 34. I've met some people, been very lucky to meet some people who have looked at drugs in hundreds of different indications, and they get insights because they have such broad knowledge, and I hope to be like that when I'm older.
So it's like the Warren Buffett thing where he just
Yeah.
reads these prospectuses.
Well, he just
day in,
knows
day out.
about Buffett just knows about every single industry, and so that helps him with a new industry, right? And it gives you an advantage because you've seen so many things happen. So you can be better than the expert in a disease because you know that disease not as well as the expert, but 100 different diseases and what's happened with how drugs have played out over time. And so I know. One guy who I actually just spent time with this week. I spent a day with him, and he's a professor, and but he's involved with a lot of biotech companies, and he just knows the history of hundreds and hundreds of drugs, and he's been intimately involved with designing the clinical trials and all this stuff, mostly in endocrinology and metabolic, and I hope to be like that. So just gradually snowballing your domain expertise over decades, I think creates a big advantage. So that's my goal, and that's what I would recommend.
Awesome. Well, thank you so much for joining us. We really appreciate it. This has been great. And
before we go, I'd like to wish our host a happy birthday.
Oh, happy birthday, David!
Thank
Birthday.
you. Thank you, guys.
Pleasure
Thank
to
you.
be here with you on your birthday.
Appreciate it. Well, let's go celebrate.
Let's do
All
it.
right.
All right. Thanks.